No problem, sir. Okay, please.
So let me share my slide. And before that, I would like a very good morning to all the participants and faculties and my dear colleagues. And I think it is a very excited program since yesterday. I am hearing that it is very much like active participants are like, going on from various, like particularly faculties and other research scholars. So I think it is like, all the topics since yesterday, the people, the resource peoples, those who are explaining, they are giving a lecture. It is a more relevant and high significant for you guys to develop in future. Even for me also, I'm just learner, right? So actually, after my talk, probably you might be heard about synthetic biology for drug engineering by Professor Joseph Selwyn from Pondicherry University, as he is one of my good collaborator and particularly in metagenome work. He is also one of the pioneer in India. So I hope you got it a very clear picture about how synthetic biology is going to be benefiting for, like, drug targets or drug engineering. And again, there is another related to my area, genome editing in microbiome. It is by Prakash, So it is one of my area of interest, which relate to the yesterday's metagenome, particularly in microorganism from various drug discoveries and from natural resources. And I hope today morning you might be enjoyed, like, how the genetics of cancer, what kind of things, understanding of genetics. That's also very, very important before whatever you are going to go for this cancer therapy or drug discovery. So I'm sure that you might be enjoyed and you got huge, like, the broader information, knowledge from yesterday. So today I would like to go towards how, like, a drug which is already used for some other diseases need to be used, like, repurposing. That's a very important, because many people, many researchers are discovering a huge number of metabolites and any proteins or any kind of, antibiotics, whatever yesterday we have discussed. But it is not reaching. Mostly it is a kind of, clinical, various other processes to implement is as a drug. Probably some like, it may be a kind of, the scientific and other things are required to develop on those kind of, novel drugs. And probably, there are certain drugs is already FDA approved drugs by WHO and other things, and each and every diseases which we are using, and like, it is like, a drug library. But though after some time it is going to be not effective on that particular disease target, those drug itself, after 4 or 5 years, if you use the same drug for that particular disease or cancer, it may not be effectively working. And we cannot simply throw them. And though much huge process which we have made, and so much time-taking process, scientific community in association of various industries are really, put a lot of effort, lot of money, and then after that it is simply lying on a kind of a drug library. So the drug library lying like so many thousands of drugs so far, which is not effective what purpose which already been reported. So for that, and today I would like to drive you in the different corner, and it can be very much helpful for you to use, even though as we know, this pandemic situation, we are unable to do in the laboratory condition. Even you can use such computational tools for drug already been available drugs, and you can use for the repurposing and various other diseases, including even your spike protein targets, COVID targets also. So I hope I will just show you the slides so that you will get a clear picture what I mean to say another 45 minutes. And hope you may have any doubts, please ask and discuss about that, we will happy to explain you guys. Okay, so let me share my slides right now. Yeah, let anyone can respond that whether my slides are visible or not.
It's visible, sir.
Yes, yes. Is it now okay, the slideshow?
Yes, sir.
Yes, sir. So dear participants and dear colleagues and friends, as yeah, I just brief about what really the drug repurposing and what could be the benefits, how need to be overcome for these therapeutic bottlenecks by using this drug repurposing. Actually, what is the drug repurposing meaning, very clearly now. So is already known drug which can be used for other things, but it need to be used as a strategy for developing a new user for whatever the approved drug or investigated drug so far available in these various particularly FDA approved drugs. And we need to be make that a specific original medical indications. That means, for example, there are some flavonoids, or there are some alkaloids, or there are some cardioglycosides, there are some terpenes, or various natural resources, or even synthetic resources, molecules are used various other purposes in well in advance. And though it is already used various disease, but unfortunately there may be a side effect and various other problems. Most of the drugs are not efficaciously showing or making that activity of the inhibiting of the drug targets. So probably you might be recommend certain the drugs which you are hypothetically, and you can find certain drug candidate which already used. You can use the repurposing, even the computational tools. I will give you what kind of a computational tools which we are going to be used, even though people you might be knowing very well about docking studies, molecular docking studies, and simulation studies. Not only those studies, and there are well advanced tools is available nowadays, and we can able to discover in computational drug targets. So in some, I will talk some little bit in my laboratory discoveries also in the papers, and I can give you kind of a hint about how we need to be performed for drug repurposing. As I have told you that what are the advantages of drug repurposing and how this is going to be helpful for various other things, but particularly mostly considerable the cutting edge research, particularly in the for example in the industries, and it is though we no need to be process of from the beginning. It is already know about a complete property of particular drug, and even cost less amount. So cost effective less means we can reduce very little amount you can be able to discover, and we can go directly instead of going phase 1, phase 2, phase 3, because we know the properties, physicochemical properties and function structure of that molecule or compound or drug. So it can be used by various things. So this potentiality of reuses are despite whatever the evidences which we are adverse effect which people are making. So it definitely going to be indicate that how this repurposing can help you, and for particularly in cancer therapeutics, even not only the cancer therapy, various other the disease targets, and as I explained you very well. Yeah, so I'm going to be speaking more in the cancer related things. You know what is the cancer, right? So it's a kind of a group. It is not just one particular kind of organ it is affected, but though it is a group of disease where hereby the cells were going beyond their normal conditions. That means the cell grow beyond their limits and invade the adjoining part of the body. That means one organ to another organ, it can be spreaded slowly, then it can be harm for that the normal function of the normal organs. So as you know that this is the data which I am presenting in 2020 by WHO reports, and there are various tools, means people are statistically presenting, most of the people, by using but cancer, the death rate in worldwide causing the 10 million death, even increasing. Even this 2020 itself, 10 million death is because of cancer. Many people living their life actually without any proper medication. So actually, and the statistics, if you see over here, and worldwide things, but mostly that the diseases are causing cancer, particularly in the lung cancer and breast cancer, liver cancers. So these are the major the cancers which are very much aggressively happening. Even right now, right, the lung cancer, obviously right now we don't say directly lung cancer by this COVID infection, because it spoiled the complete normal whatever the breathing, what is happening, because it's a heart of complete without breath you cannot. But followed by the breast cancer and liver cancers. So where the statistics suggested that here in India it is predominant by breast cancer. I don't know why, what could be the reason the people are getting from Indian scenario. It is having that breast cancer, it is in top. So, and if you see that individual the countries, indication of this the colors, most of the people diseases, lung cancer 87% people are death, and breast cancer 30%. So in if you look into that the percentage of death in the worldwide, that indicates that even it is worse than the COVID, and this is not statistically reported by, and people might not be aware of about anything. So you can see that Indian statistics, the ratio by WHO is given in 2020, and as I said you, this breast cancers occupied is a top one rank. I don't know. That means that whatever our habits or food habits and whatever other activities might not be the up to that, because I heard in the beginning, like 2018 and 18 and 17, that almost in the one decade, probably the Western countries are having the highest the disease, in particular in the breast cancer. But I don't know how the India had this issue, but that need to be investigated further. And the the breast cancer, then cervical cancer in the second rank, it is occupied by the India. That means the people unaware are many diseases. There are statistically there are many. I don't want it to be go in detail, but I can say that how lacunas we have are hygienic and whatever the maintenance and health related things, we are very much neglective. So in that scenario, so we need to be study and investigate and various other targets. So in that way, as you know very well, the breast cancer disease, which people you might be very well known, certain drugs are already well reported, particularly certain the gene targets WIM, and that name of the drugs are breast cancer drugs, Vimlistin, vincristins are also is well-known drugs, but they are not now effectively working or effectively functioning to prevent this abnormal growth of the breast tissues or other things, even cervical and various other. One by one, I just wanted to explain you for your detailed things. But in order to make you that why we need to design such a drug development process, and whether it is already drugs is there, then why we need to be again reinvestigate them. So that definite questions will come to the people, So the main hurdles of the various cancer development is that probably you might be noticed so many other issues, but regulatory perspectives, particularly you can say scientific discoveries. The scientific discoveries mostly it is not reaching to the people or reaching to the drug where the people are discovering up to the end of to the industry sales or the medication. But even the clinical, so many hurdles, Yeah, it is not the process of I have discovered a drug, it can go by another 2-3 years for the clinical things, and it can be applicable, but it is not is happening in the scientific community. And lack of incentives, incentives are outside the companies. That means the companies are not encouraged various other to have the government of various governments are not much focusing to encourage the companies and even researchers, particularly the basic, even as you know that our GDP, the scientific contribution is just a 0.2% than compared to other states. Nothing in front of our budget actually. The government need to be look into the health issues now. The COVID, how much it is, the pandemics made, and how many million people we are lost in front of us himself. So that need to be enhanced. But the main thing is that the government or something need to be have a regulatory conservatisms. Means the conservatisms means it need to be a kind of a statistical way and cultural and social and socio-economical based things need to be changed, and politically need to be changed to develop this conservatisms, and need to be bring that idealism, ideologies. So the ideologies very well. So since many states we are not that much 100% reaching the literacy, through the literacy also we can able to know various ideologies and we can get the things, but it is not happening in the Indian scenario, but need to be improved. So the coming days it is already going to be improved, and slowly we will see that how it can be go. So non-harmonized regulatory views, that's what I say that. So that means the ideologies are not that much making the regulatory authorities, like for example IPR sections or kind of intellectual properties or something, various other things are not strong enough to develop and validate these drugs. And certain techniques is very important. You have your aim to do something to new discoveries, but if you don't have nearby techniques, tools to how to develop this particular molecule in form of a drug, that is very much lacking in this, even though pharmaceutical productions are very low as compared to Western countries actually. So if you look into that here, how the statistical way need to be go, the target hypothesized and target identification of whatever the function and whatever the things, even we can say, for example, in genome based also, which I have even yesterday we have discussed about genome based drug targets or something, and that also can be easily hypothesized, and we can go for various compound screening, and then particularly the preclinical test, and then find out a specific lead compound and a specific disease, or sometimes it is a multifunctional or a kind of unifunctional, also no issue about this kind of drug targets. And obviously very well, and the phase trials, and there are huge hurdles over here, and this all, if it is going to each one by one, almost 90% drugs are just failed on those trials actually. And then obviously it goes to the level 3, and obviously it goes to the medication or a particular cells of that particular clinical use or particular things. That means a kind of either you can go a conventional method, or you can go for the advanced technologies which we can use and find out the drug target and particular target and development, and use the clinical uses. So this all we can say, for example, one way is a molecule based targets. Even there are many other targets which people are working, and yeah, of course this is repositioning also we can refer this repurposing. Repositioning means is a kind of a engineering of that molecule, can be make it a unique function, or the strategy where it is existing the drug leveragated in the new targets, a new say for example a kind of a foreign a kind of substance or something, a atom or something, a bond in the chemical structure, you can able to remodify or something, go for the various green chemistry targets also can be used. And for that, and we can make it a lower risk of failures. The time framing of the drug can be reduced, and sustainability or something, it can be required the less investment. So that these cartoons can see that how this people are going, and then once it is approved drugs and for other diseases and other some X diseases can also be used, and various changes or modifications are genetically and not genetic, chemically engineering or something we can use for the drug repositioning or repurposing also. Okay, yeah. So these are the trials. Now you are going to be have steps to before in your mind to discover or something what that means, what kind of things I wanted to do and why I wanted to do and how I can perform this. So probably I may not going to go in a gen, and I can go a kind of which my lab is going to be focusing on certain molecules called cardioglycosides. So the cardioglycosides, which ancestrally, so since it is a plant derivative or secondary metabolites, and it is using for heart diseases, initially in the 90s and 80s, it was using the heart failures targets, but none other than other targets, they were not reported so far. And when the moment, if you see that the trial watches, and started in the just in 1950s onwards, and this work is started to make that cancer therapy also. So that means you can say one of the example, digoxin and various other unlocks, people are using the novel cancer therapeutics because various other things. And anti-cancer, whatever the immunogenetic modulators or property modulators, and the repurposing approved or whatever. But most of the drugs in the repurposing which the cardioglycosides used, it is FDA approved. So, and few are drugs from the cardioglycosides where they're using these various other purposes, but need to be validated. Still, it is in lacuna. So for that, my lab is focusing towards various the cancers breast cancer, liver cancer, and lung cancer, even cervical cancer. So we have been publishing since a decade about these cardioglycosides extensively, and we are getting high responses and about to various other things. So that's the reason it is my one the interest on this drug repurposing to develop for the society. Okay, so the time frames, if you say that if you look into that, the drug development process in the casually I have in the previous slide I have expressed you how much time it is taking, and there are so many the bases so many years it need to be almost 15 years it takes to go to the first trial to the clinical trial, first phase to the clinical trial. So if you look into that de novo drug discovery development targets, and obviously it is a low success rate sometime, but it can be make it in a huge the price you can save it, and then time also we can save it. So that means if you know that whatever the toxicity or about the drug, the purpose of using some other things, then we can easily can find out where this safety of the drug and the various pharmacokinetics based studies, pharmacogenomics and pharmacokinetics, pharmacodynamics or various other approaches we can use and go for the drug repurposing. So if you see the traditional approaches in this picture, so how difficult it is in the face in this box, the left side box, and it indicates that, as I told you, that the identity of that particular therapeutic target in the disease interest, that means every researcher cannot be focused on many kind of disease, right? Or something, one particular interest they may show, like breast cancer or liver cancer or whatever, the lung cancer. So then if you look into the second step, developing the potential drug candidate means a laborious screening required. So the laborious screening, is a kind of, it is, though it is robotic screening, but man, human errors are many, and to need to be initially go for the cell culture assays and toxicity assays, and followed by animal testing. Directly we can treat them in vivo studies, and then find them to whether it is going to be how much it is get success. The cell culture activity might not be a directly, animal directly cannot be work. So there, if this the ratio or something, the efficacy need to be built various dose dependent manner, and those successful targets, then definitely we can go for the clinical trials. Like that, there are so many steps. The phase 1 of, though this is itself a big task, it takes almost 5 to 8 years, and then phase 1 and go for the safety and whatever the things and effectiveness. And though is something going to be in the failures will be there. Ineffective means obviously failure, it never go for a second phase, and like that. And then sometime it is if some side effect will be there, it may not be going to go in third phase, and some 1 or 2 only it is going to become as a phase 3 trial. And then, so that too, this is a high cost effective also, though it is a traditional approach, and 10 to 15 years it will take. But you just look into that here in the right side box, and straight away, since we all studied about hurdles, various steps, then easily we can move straight away for the repositioning or repurposing. So for that, and the safety and efficacy, we all prior information we know about particular drug, and then easily can go for the treatment and clinical trials. And in that success rate will be very high, like a phase 2 and phase 3, and within 2 to 3 or 5 years, before 3 years, it easily can come to a clinical trials. And that too, it is very less price, and the time we can save. At the same time, we can able to money also, we can save the money and time, and easily can go for the clinical trials. So this is the one benefit of this, instead of going to a traditional way to the drug repurposing. So even though today, which we are using the vaccination by from the COVID, it is all repurposing only we are using. So they are purpose of somebody else which we are used, and the same viral proteins we are going to be having a various the vaccination. That is the reason it came within a 6 months of the approval. Otherwise, the regulatory the FDA, it may not be going to be, because if something went wrong, the entire population we are going to lose. So for that, the repurposing nowadays is very much hot areas to discover and make a very reasonable targets or reasonable benefit for the society. Yeah, so maybe you might be knowing with the computational approaches. So the computational approaches we can use individually or something combinatorial, the combination of systematically X and Y, X in some other purposes and Y in other purposes, that we can make it in engineer that, and we can go for the large scale production and go individual targets, individual targets, a genetic association, how this genetic association are going to be related to various the diseases or something. Morning you might be heard a lecture on how the genetics the understanding of the genetics can be. That means a genotypic within a population, it co-occur the phenotypic traits, that the phenotypic traits often than would be the expected whatever the chances is occurrence in genetics kind of things. So in that way, so a genetic association studies, I mean a genome wide association or genetic association, it gives a kind of a particular sequence on that particular allele or particular gene or halotype, we can say, in the chromosome, and involvement of this controlling the phenotype of a specific trait and metabolic pathways and other diseases also, right? The pathways we can network studies we can do. And before that, as we are beginners, basically we'll try to do various trial and error method where this molecular docking studies and everyone can perform here, and that it is the most frequently people are using, and the structure-based drug design due to its ability to predict their binding conformation or their binding efficacy with ligand and protein interactions or whatever the molecules is happening in appropriate targeted binding site. Okay, so that people are using, like, and going for this molecular simulation and other things. And coming example, like in order to understand the pathway mapping, how this mapping can use the genetics targets and proteins or whatever the relations, and it is obviously we need to understand about network system the pathway, we can say. So in that pathway, probably you might be knowing straight away we can go a KEGG pathway mapping, and then we can go for the various experimental evidences in a specific targeted disease or in an organism, and then we can go through even through genome sequence targets also we can target, and even a specific target also we can go ahead for this thing. And you just I hope you might be knowing a systematic analysis of whatever the reproductive clinical analysis. There are nowadays people are suffering with retrospective targets. The retrospective targets where the people even they are taking their own medication, and that is not good, and that is not recommended by these doctors. The data whatever which you have are something to gather and them to make a repository, and that's what every the clinicians are going to be analyzing systematically. Then they will going to be give a clinical medication. But still, the researchers are focusing on these retrospective clinical targets and analysis, how it can be helpful for things, and find out. I told you please in vivo and in vitro drug screening. So through this, and probably as my lab is focused on genome based data, and that drug banks which we use are we are taking that already available drugs, and the sources I will show you the later slides what are the databases which we are going to be used it. And the phenotypic screening very well, and the screening whatever the biological activities or drug discoveries people are using so far, the identifying the substances on that their individual small molecule to the peptide or something a bigger molecule. Even there are certain RNA targets also can be done by the phenotypic of the cell and organism or whatever the desired targets. So the screening involved probably you might be understand that pathway. I told you the previous one. The main component of the biological pathways to identify these phenotypic targets. For example, stem cell derived human cells. So how this is going to be working? Then obviously we can individually isolate them, individual tissues, and go for the the sequence target, or even that specific conventional target also, or even we can go for a complete organism or the whole genome, I can say, and complexity or complex based screening we can able to make it. And finally the signal based match, how these mechanism or pathways are interacting each other, like one drug for one disease, but it might not be, and one drug that it may be having a side effect or something other disease also. If you target in the breast cancer, probably it may be going to be sometime it going to benefit for the other diseases also, other cancers also. But this binding assays need to be identified, and one drug for multiple targets. So such information or such kind of targets, by using the genetic cancers identification by the genome target also, we can go for the real discoveries. And as this is a recent one which people recently published one of the good journal, and they have found how this normal cell behave and how the abnormal cells behave in the pathways or something. But the hallmark of this targeting to this abnormal growth and various the combinatorial targets, I can say, for example, and sustaining the proliferative signaling molecules. That means a kind of individual called monotherapy, and the combination called combinatorial therapy, or combination or combinatorial, we can say. So, but monotherapy might not be always going to be persist. That individually, sometimes it is going to be have a good effect, but it may not. But in the combinatorial, or I can say a synergetic targets, it definitely going to be supported and to target those things. That these are the certain drugs I may not. These people are using monotherapy and combinatorial therapy so that nowadays the combinatorial therapy is very much having the activity towards multi drug targets, or multi kind of making what we can say the inhibition of this the abnormal growth of the cancer cell, or various other pathways to inhibit that signaling pathways. Okay, so and you need to be know the what really the strategies of this. Okay, that you can see that whatever the drug discovery and validation we have spoken the previously in a different manner, and if you go into that what the current trend people are using the drug discovery, like a known knowledge based drugs, because how the library we drug repurpose means DR, doctor, so that is one thing. Activity based drug targets, drug repurposing, and which we already found this, and we have seen that various steps involved in this. But you can see over here in silico DR, that means as you know that since yesterday I keep telling, we are not able to do an experimentation and validation, and you can try to do. These are the tools and the preliminary studies for in your home itself, and obviously molecular docking. And if you have some kind of transcriptional signal targets or something, pathways, signal transduction, if you have in your mind that this is the disease having this kind of certain genes or plays a crucial role, then we can find that signature gene or signature protein, and then go for the how they are networking relations with other signaling pathways, and we find data mining and machine learning. Yeah, this is the one of the very important nowadays discover, many aspects or many research and is helping nowadays this artificial intelligence and machine learning or deep learning system easily can identify and to make them to learn the mission, and is obviously can give you in front of you in a second itself, instead of going a traditional way to search in the literature. And obviously you need to be validated now. Every aspects need to be go for the lab condition. There is no exception of this. And retrospective studies means whatever the either these all the the genetic based and the genome based and whatever the targets which we have, and obviously we have done this kind of meta analysis and taking some molecules or proteins and docking studies. So up to here in silico can use and find out through in silico transcriptional signals or pathways or signal transduction pathways. Then it comes to the real validation in vitro studies. So this everyone knows you that first starting either normal cells and cancer cells. So we need to be validated normal and cancer cells whether this drug is effective on cancer on normal also. If it is not in the normal showing that any inhibition, I'm sure that it is going to be very active compound for the abnormal cells. Yeah. So the similar way you can go for the knockdown gene found any specific pathway targeted, and definitely it can be used in vitro, and need to be seen a kind of in vivo analysis, particularly ultimately need to go for the animal model, and various targets are there. Just example is genografting or something where it's dose dependent manner, and we can go for one organ and to the various organs to identify how this can be useful for affecting the drug is going to be having the active role and various other things. So that's the reason this all we cannot like exceptional to straight away I can go for the drug in vitro and in vivo might be not significant, because we need to study about like what is the kind of the importance of the drugs and previously used, and what is that safety and other all computational network targets we can able to identify them. That's the reason the drug repurposing, the doctor can help us to save a lot of money and lot of time also. Yeah, so this is the one another publication recently. It was published in last year, and screening the therapeutic targets and drug repurposing of the cancers. So that means as we are speaking about experimental uses of drug repurposing, like I told you, the phenotypic screening and the binding screening, which the previously, but these all in olden days or something, because traditionally people is going to be do that. But we need to know about a complete a scientific drug repurposing. So it is not just if a cold came and something we are using a turmeric or something kind of ginger or something, but we sometimes it is okay, but in the raw science or some vague science, but need to be validated a computational one, so that you can easily identify even though the real world database analysis also. So that real based database is also going to be give you what are the drugs are available in the clinical trial level 2 and 3. So there real world data. So this is also free access is there. Only need to be registered this RWD tools or site. Then it can go for validation whatever you are going to be. And obviously very well this chemotherapy radiation everyone using. So unbiased targets, people are very much believing earlier days, because there is no other choice in the emergency case. But in the targeted therapy, that's very important. The specific mono-targeted drugs, it is already show some kind of high effect selectivity or high efficacy of this thing. So in that maybe that I told in the previous slides, and how this selective molecule can be used for other purposes, multi targeted purposes. So there it can give you the new name as a comprehensive therapy, drug repurposing therapy. It is not such as such, so that need to be have a comprehensive therapy target. So that now this 2020 and people are very much showed interest to have a comprehensive therapy by using drug repurposing. Okay, so as you might be having, I also have so many questions on this, but is drug repurposing worth the whatever the efforts which we are? Of course, we are clearly understanding about that particular molecule, safety and structure and function, and whatever the pathways which some reports are there, particularly a selective molecules or selective drug targets, and it might sometime it might not be unexplored, even some diseases or pathogen causing various progressions of various things. This information which I have already published last week, there is a frontiers in immunology about COVID spike protein, not the drug particularly in cardioglycosides. Although very well the marketing and the importance and the potential targets of these all things, really it can be helpful, because since our scientific community might not be so active to reach this molecule, a selective molecule to the clinical trial, because the huge expenses required and the industry collaborations required. We cannot go in the academic researcher or even a scientific researcher, even national importance institutions also struggling to have, even ICMR is struggling to have certain companies to get these challenges. So that's the reason the scientific, the regulatory things I already told you in the first slide, and it need to be a scientific perspective targets. As I already just told you that how the important, so we can build them the repurposing. So in this way, the main thing is that we can save the time and we can save the money and we can make a quick response or quick targets of your hypothetical things, so that you can easily screen them and find out a best lead drug for one particular disease, or even multi particular diseases also we can able to identify. So that reason it is really worth enough for using the drug repurposing. Then what are the challenges which you people are facing, particularly we are facing? So we know about the advantages, and we know how this going to be worked out, like demonstrated anti-cancer, whatever the acceptable safety and tolerability, and whatever then directly it goes to the human. And there is still difficulties in the later stage of a clinical trial because of the competition from the successful new drug development, So the competition obviously will be there, because you need to go for some patent and patent rights through that, and you can approach certain industries and go for the specific memorandum of understanding. Then you can easily. But in case of if something it is a legal regulatory, something happened wrong, then obviously it is very difficult. That's what the problems in this. So in that we can directly straight away go for the patient the patent consideration, and after that we can go for this inequitable prescription of whatever the pharma industries are having their own rules and regulations, and then we can explain them, and drug we can definitely promote to marketing to the physician or whatever the drugs the doctors people are using of various targets. Yeah, so as I promise you that how like the cardioglycosides so far used various drug repurposing. Not only that, example I just took, for example, a digitoxin. It is already, I told you, congestive heart in the ancestral days, and now it is not only this is highlighted. Digitoxin is showing various other cancers inhibition, like lung and pancreatic, leukemia, and breast cancers. And not only this, even a 16 number of diseases, a single drug is having targets. This is recently in 2019, FDA approved digitoxin is one of the cardioglycosides. And the other one is, though it is relaxifin and other purposes, but this is using for the bone diseases. But now you can see that the cancer, breast cancer, we can use for using drug repurposing. Aspirin, very well, and we use without even medication, doctor prescription, directly we can use a pain reliever or something. But now the people are using the same aspirin for the colorectal cancer. So that means at least are many, but I have given a few only. But there are a huge library is available in the drug repurposing and drug databases. Okay, so in that manner, and I just wanted to say how this cardioglycosides, because I'm more familiar about cardioglycosides, though it is highly complex structures, it is not so easy to have characterize this, unless we take the help of the chemistry people, So this relation, there are a different combination of these molecules. So the different combination means a saccharide moiety or will conjugation with the glycosidic linkage, and there are various methods in the linkages, 1,4 and alpha N glycoside linkage and other things, which whatever our ribonucleic acid structures are happening, the similar way it is going to be. And finally, and if you see that steroidic moieties. So the steroid moieties means a kind of a hormonal based or a kind of drug, particularly even cholesterol based molecule, and which is associated with the lactone moiety. So that means this much combination, a single molecule which is having such a huge the importance, and by structurally itself, a structural diversity itself, you can able to know how this sometime obviously I told you that this is using for the cardiac arrhythmia or something, cardiac failures, heart failures. And the real function of these cardioglycosides are going to be used in that, like, sodium potassium pump, then while pumping, like through the ATPase things, it can be disrupt the membrane ability and then to go for the tumor. So this way the cardioglycosides are targeting or something. That's what it is, a simple method, and where it can be easily we can target, and which moiety is going to be working, that we need to be easily can identify. But if sometime a permutation and combination also we have tried, like, removing this steroidic saccharide moiety, and and a combination of this steroid and lactone moiety, and vice versa. If you check it, and there is only the high efficacy people because we showed even with this combination, that means the three combinations. So these are the drugs which we are using for the repurposing in our laboratory, and it shows that huge multi-targeted diseases. As I told you, I am using 5 diseases in my laboratory by using the cell culture method, particularly not in vivo and in vitro study itself. So I can go for the targeting the cytotoxic activities and various other signaling pathways, and all the things in which have, just for your clear understanding, I just wanted to give you because it's not having a color picture. And since we use MCF7 cell lines, breast cancer cell lines, and even the lung cancer A549, and the liver cancer HepG2 cancers we use, and we use one of the perovskite which in the list, and the streptomycin and lanreotide. So these are 3 drugs are after screening I got in the huge potential activities of this, and I just was in 0 to means control to the 24 hours and 48 hours. So in the moment the cancer cells, if increasing the time in the increasing dose, we have tried to dose dependent also, but I'm not showing these cytotoxic things, but I'm just the image. I'm just wanted to just make you clear understanding. That means in the 48 hours, this perovskite is completely killing, almost 80% kill this cancer cells. That, similarly in streptomycin and lanreotides and other cell lines also. So that indicates that a preliminary understanding about how really these cells, these molecules are effective on various cancers. So this have been published, so many publications. So then we need to be understand that's what that pathway targets, right? So this is the the hypothetical pathway where exactly and this all published which by my team, and we used, and there are a direct key inhibitor of various signaling pathways, and finally there is one thing which the CD47 and HIF alpha, hypoxy, the things which can be directly inhibition, but there is no report so far, and this can be directly goes to the autophagocytic system, and that means that macrophages can be engulfed. So that also the people working on these pathways, and this is a new pathway which we have discovered, and it has been published in the various other Frontiers Oncology Journal and other journals, cancer journals. And this indicates that in vivo, in vitro study itself, we can go, that we need to be validated further all the various molecular tools. Yeah, these are the the journals which the 3 molecules are published in targeting individual targets, individual signaling pathway based, or some various other pathways which very much helping to identify that, but still it is lacking to go for to other in vitro studies, but still we are establishing the in vitro study laboratories. And you know that how the cell death modulators can work these cardioglycosides. So this is our hypothesis, like, the immunogenic modulators. So we already, our papers in the review articles are biomolecules and drug discovery today. It has been published last year about this a complete immunogenic modulators, like immune modulators. So there we can say that, for example, the activation of immune cells, and then you can target individual mechanisms or individual key genes where they are really inhibiting in that particular the cells, in the kind of targets. So that huge things are there, but I am not explaining individual things, how it's going to be work it out. And obviously, you might be just now understood about drug repurposing methods. Probably the step one to this new drug. That means we can call as a same repurposed drug. If something it is no report so far, or something it is having some other disease target, we can also call as a new drug by the DR. DR means drug repurposing system. So that means all the process without escaping one step, the learning and evolution and drug disease prediction and structure and function. So obviously it goes to a common man. So this is one of a pipeline people used to do that. And yes, I just promise you that these are the networking tools people use, drug disease networking reconstruction, and see the DRGN and DIGN. These are networking things we can easily find out, and then use by this data bank. I mean, the source of data banks, drug bank and CDT, and there are many. So then you can find out in each drug bank how many molecules are available and how many are in the known and unknown, and then their encoding genes are even the particular gene bank. Through gene bank also we can able to identify those things. So you can use these are the URL IDs. It's all free access, and easily you can hypothesize your targets, and you can find out what are your targets by using already used drugs. By there are drug libraries are many available, and even FDA also is many things are available. But now these computational approaches are a drug which I just showed you previous slide, that what are the database and what are the sources available. But sometimes conventionally doing is okay, but it is a time taking again. But if you make them to those drugs or those genes, and you make to mission to learn, that means it is automatically divide and give you in front of in front of you the molecules, whatever you are targeted. So that's the reason. So that machine learning targets nowadays is very much robotically using, and is a promising tool for whatever the to drug you need to be accelerated, and it is very, very significant also. And you can filter see 10,000 drugs to and screen a very minor or something kind of. So for that, the individual steps and prediction of the aim of identity and their potency, bioactivity, toxicity, all the things with you feed them in a database, and then look into that virtual screening. That's very important before. That's what molecular docking also we can as one of the virtual screening. And we are doing virtual presentation, virtual learning. So that gives us a target identification and predict them, and to identify the targeted whatever the upstream targets proteins and downstream target proteins or something. So the similar steps is involved here. This is just copied from the literature, how this preclinical and this individual virtual screening going to be happen. So then the deep representation of learning, that means the machine learning, and gives you a kind of a individual molecules are classified the molecules, and that the drug targets, the mission can always fed it and easy to make representation through the machine learning. The artificial intelligence, that's what we can say, this artificial intelligence. So nowadays, very well, it is increasing the effectiveness of purposely for this drug repurposing. Again, I'm telling you, this is one of the big scientists published in Nature, and it says that this is really very much helpful for know about the available drugs. Instead of searching conventionally or manually, you are searching for individual one drug to another drug. If you are available the machine learning targets, and automatically the machine will give you or brief you, and this drug is for this purpose, why you are using the other purpose. Like similarly, like how robotic robo is going to be explain you, the similar way it can be helpful for the society, and using these computational tools and machine learning tools. Yeah, so these machine learning tools, there are different approaches, might not only one particular targets, and this always going to be very authenticate. That's what I'm gonna to tell you, how genome analysis or genome-wide association studies are giving a clear-cut picture about even hereditary disease of a human or whatever things. If you just take now the Western countries are already following just a born baby, like the whole genome sequence is available, and they can predict through this machine learning. Like they will feed it in the machine learning, and when you go with the doctor, and you can just show the give, instead of seeing the face, and you can give the chip, like whatever the pen drive, and they open it and find out what would be the expected disease for you, what is that now you are suffering, where exactly it got mutated in your genome, or kind of things. So it is very much helpful for screening, for the high throughput screening. Sorry, someone is messaging, I unable to see because okay, I will respond if anything later. Okay, so in that manner, as we know that this is the pipeline with genome and transcriptome and proteome and drug targets, and individual layer screening, like input layer, and then their networking studies, how their structurally relation and functionally relation and their property. That means a structure function relation can be studied by using these artificial targets or artificial deep learning mechanism. So with that, I hope and artificial intelligence, like the deep learning system can help you simplify your approaches, or can you utilize this while sitting in home itself. We no need to go in the lab or something. Yeah, there are certain data. It was recently published, the FDA, how this computational repurposing, and it can be used for the biological networks. So biological network, obviously, this like heterogeneous biological targets and chemical biological targets, like cheminformatics or chemoinformatics, chemical informatics. So these both depict this whatever the true of drug resistant relations. So that means you see that the heat map of this FDA approved recently, and you can clearly identify the whatever the structural based drug and how this need to be simplified. Specifically, if you target in the breast cancer or something, you can see over here how the gradiently they are finding the FDA drug for this various correlation matrix. This matrix can clearly help you by the machine learning or computational targets. Yeah, so this is the one other way, the missing piece of the artificial intelligence in medicine. So what really it shows that the promise when the applied drug development, particularly as a biomarker prediction or drug repurpose. So anything we are identifying by a drug certain disease target means we can say as a marker, right? So that biomarker, we can even any kind of experiment we can use as a marker certain things. So there need to be solved, like preclinical targets, like where it is I already told you there is another way of the presentation where this convolution of the individual neural network also can be identified and validated in the cross validation, and then individually that network, I told you that the previous slide, how deep learning tree or decision tree can help you for the transferring the whatever the knowledge you are want to gain from with the data bank and whatever the databases and transfer the learning system. And here, and the scientific literature based on network based targets. So this is a kind of a psychological assumption. The people are already working scientific literature nowadays. So many so many publications are coming, but there is no use at all. How it need to be used and whatever the things that need to be targeted, if the artificial intelligence or the mission and can associate with the networking studies, all the diseases Alzheimer, Parkinson's disease, autism, so many diseases in the kind of and that you can directly the literature based the database if can be made, then easily we can target this kind of diseases. So these diseases are now very much psychologically and facing the so many issues and brain tumors and so many. Though it may not be suddenly, it can be a cancer. It can be a certain things. Is a psychological kind of things or neuron diseases. Suddenly, it may be going to be have even schizophrenia. So the brain failure because of neurons, how they are functioning or something. So this can be helpful for even drug repurposing also for this purpose. So that just I want to bring you in your notice that how we can able to use on this. So in conclusion, probably you might be understood about how the strategies which we are using for the cancer diagnosis, and obviously the molecular characterization, which we have already spoken in the various aspects, and genomics alteration, tumor mutations, and microsatellites and macrosatellites, and various other targets are there, and chromosomal instability and cell origin. So in that way, the molecular characterization can be done, and the tumor microenvironmental characterizations, that where exactly is happening, how this is happening, and what are the signaling associated, the tumor associated and neoantigenic targets are people are using. And finally, the pharmacogenomics discoveries are very much helping nowadays. That means genome-based targets like genome-wide association can give us a very clear-cut picture to target the disease drug discovery. This is the various parameters in that drug discovery, target discovery, and drug design also. Then ADMET predictions, that what are the side effects and what are the other things which can be admit things, and followed by drug repurposing also is one of the major issue that I wanted to focus here, and drug synergy, that also a combinatorial targets. So then obviously you can go for the finally a clinical outcome, and the clinical outcome where it can be stratification, individual patients, the morning you might be heard genetic counselings, genetic based cancer targets or something, and definitely it can be helpful for response to the therapy or whatever the people suffering various diseases. So with that, I may be going to conclude about my talk of overall brief information about from the straight away in the drug individual molecule to the using various the computational and genome and experimental in vitro and in vivo targets. So with that, I thank you very much for your kind attention, for listening me since almost all 45 minutes. Okay, thank you one and all.
Thank you, sir. You have enlightened the talk on cancer therapy and drug repurposing. So, dear participants, you have any queries, please unmute your mic and raise your question, please.
So, Divak Tera, that means you have raised your hand. If you have any queries, you can interact with the speaker.
Thank you, Rajesh Kumar. I hope you got it from my presentation. Some information might not be possible. Entire information you can get it, but because of lack of time, it is we need to be restrict certain things. So, if any interest, if you show any interest, I'm sure I'm happy to associate with you guys by mail and any WhatsApp or other things.
Okay,
Yeah,
sir.
please. Please. I hope you might be having many doubts. Probably, people are. Thank you, Rajesh. And hope because I don't know, but we are working on this concept in from home itself. But since one and a half, almost 2 years going to be now. By March comes since 2 years, and there is no progress of many scientific communities. And this machine learning, drug targets, and various repurposing definitely can be helpful for us. I think someone asked, let me check the chat box. Is repurposing technique also work for combinatorial dose forms? Yes. Yeah, this Akash, it is going to be work it out because, say for example, I told you in the previous slides where an individual drug, though it is the purpose of one disease, is it right? So, but sometime it may be having their efficacy may be lost, and it is simply lying on the data bank, and there is no benefit for the human. So there we can use that one repurposed drug, then screen various the networking screening, and we hypothesize if still that is not working, I'm sure that we will take another repurposing and make them to a combinatorial. That's what the combinatorial therapy is now very much working now. Even our what we can say, even COVID vaccine also is the combinatorial therapy, and the mixed vaccination. That means the cocktail vaccination nowadays is working now. And earlier, individually, they are like taking from one chimpanzee or some the human adenoviral protein, and recombinated the vaccine preparation happened. But now my query also, because one of the doctors, the famous doctor from USA, is Indian origin, and he has given the talk, and we invited him, and he the proposing like this kind of, so based on his initiative, now the mixed vaccination, I can call them, instead of mixed vaccination, I can call them a cocktail vaccine. So why we need to go every so many vaccines nowadays available, even Covishield or Covaxin are so many, Pfizer, Giger, I don't know, because of the rich people can easily take those, but like we are 75% people are very poor people in India, particularly in average below BPL people. So they cannot spare the money, So for that, if one vaccine or one drug or something with the combinatorial things, definitely it can be helpful for combinatorial drug repurposing targets. Yeah, I think Shubhajyoti had some chemotherapy, target therapy, and comprehensive therapy are implicated after detection of the cancer. Yes, of course. And what you would like to say something? Like we vaccine, yeah, of course. I think you are explaining. Is there any questions? Yeah, the type of that type, is there any technique which help us to prevent cancer? Yeah, it can be understand about the cancer. Definitely that whatever the computational or bioanalytical techniques are really going to be detect certain abnormal growth of cells. Like yesterday someone told, we can directly target. Nowadays is a single cell live image analysis, how the single cell is going to be behave. If a normal cell and abnormal cell, if you observe them, and the entire entity, the pathway is different from the normal cell by this abnormal cell. So that definitely, maybe I don't know which technique it will directly prevent it. I don't think so, but we need to be work it out with all the evidences, all the technical evidences, we can able to go for the clinical trials. So that's what I can say, Abhijyoti. Thank you. Thank you. Any questions?
Any queries from participants? Yeah, okay, sir. Thank you a lot. So, on behalf of organizing committee, once again we thank you, sir, for your enlightened talk on cancer therapy and repurposing drug aspects. So, thank you lots. So, dear participants, so kindly be rejoin at 2:15 p.m. on the third session will be start. So now,
I
thank you
would
all.
like to, yeah, thank you, sir. I would like to request to participants,
Yes.
if you have any a kind of, doubts, even we need to be associated in the collaboration in like, various like virtual collaboration, even experimental collaboration. I always welcome you. I think in my first slide, there are my email IDs. You can try to mail me and reach me so that we will think about any such association and means the collaboration of any kind of work on computational or even artificial intelligence, even experimental. So we are looking broad collaboration to everyone. Even we associate many universities abroad, and I have 5 countries collaborations. With the help of them, I am reaching and each and every technical oriented, and even I am not expert certain areas, but they are helping us to support our publications and our projects also. Thank once and all for listening patiently, and I hope you have got some information about from my talk. And I thank you organizers, especially Sanmugara sir, and thanks for giving me this 2-day program, since it is a very hectic for me because I have classes even in between. So I have done, and successfully even exams are going on. So I'm alternatively going the exam right now. I shared before while you are talking the question paper and they are writing now. So that way the hectic things. And even though because our honorable vice chancellor invited me today by meet me by 10:30, then I denied because of this FDP program. I have the lecture. So after that only I can come and meet you, sir. Like that, I took permission from the vice chancellor. Thank you very much, and thanks for giving me opportunity once again. Thank you so much.
Thank you, Professor.
I think organizer can add a group, a WhatsApp group, so that it will be very much helpful for interaction with the resource persons. Sanmugara, sir.
Thank you, sir. So already we have the group. We are sharing all the information with the group. Thank you for
Yeah,
your
please,
effort and support, sir.
please add me in the WhatsApp. And even Rajesh also is asking. I think he has shared the number. My number also you could please add in the WhatsApp group. So it will be
Okay, sure. I will add you, sir.
not only this for FDP program, in any kind of thing. We are for the society. We need to be support our people. Okay.
Okay.
Thank you so much. Thank you.
Sure,
Thank
sure.
you.
Thank you, sir. Thank you a lot.
Yeah, with your permission, I may be exiting
Okay,
now.
sir. Okay, sir.
Thank you so much. Thank you.
Thank you. Thank you. So, dear participants, please rejoin at 2:15 p.m. So now today the morning session is over. The afternoon session will be starts by 2:15 p.m. Thank you. Thank you all.